This program is brought to you by Stanford University. Please visit us at stanford.edu.
Let me begin with but a few of the highlights from last week's lecture by Professor Daniel Dennett of philosophy at Tufts University, the author of our course reading, Darwin's Dangerous Idea. One of Professor Dennett's main points last week was to highlight a key insight of the Darwinian paradigm, the emergence of adaptive, even intricate designs by so-called mindless, purposeless forces in nature. Darwin and Wallace had, in natural selection, a mechanism that fully explained the appearance of design without supernatural designers. Turning on its head the work of an early Darwin critic named Robert Mackenzie, Dennett applauded Darwin for the, quote, strange inversion of reasoning by which absolute ignorance is fully qualified to take the place of absolute wisdom in all the achievements of creative skill, unquote. Says Dennett, Darwin replaced that whole top-down view of the cosmos, that whole top-down causal pyramid of God the designer, with a bottom-up designer, the process of natural selection. In order to make a good caddisfly food sieve, Dennett argued, it is not necessary to know how to make it. To be very effective at pushing eggs out of a chick's nest, says Dennett, it is not requisite for the cuckoo to understand what it's doing, and we had a graphic example. These are among the myriad wonderful adaptations shaped by the infamous blind watchmaker, natural selection. They emerge through the survival and reproductive advantages they convey. Again, following Dennett, take the eukaryotic revolution, for example, when one single-celled prokaryote invaded another prokaryote and thus incorporated what Dennett called 2 billion years of R&D. It was a stunning and successful transformation in the history of life. In his book, Darwin's Dangerous Idea, Dennett argues that big transformations like this in the history of life don't happen through what he terms skyhooks. That is, interventions from supernatural agency, no skyhooks. Rather, they come from major evolved innovations of structure or function that, quote, kick organization up to a new level, as he put it last week, which Dennett calls cranes, construction cranes. But as with all cranes, these work from the ground up. There's nothing supernatural about them. The advent of sexual reproduction in living organisms would be another construction crane in Dennett's terminology. Dennett's second big point was that human culture, too, is another such major innovation, another crane. We are, quote, apes with infected brains, as he put it, infected with ideas that we have acquired socially from other brains. Culture thus offers us a second information highway, as he put it, and more than that, it gives us a second track in which evolution occurs, a cultural or mimetic track of evolution. A meme is therefore analogous to a gene, a replicable unit of information that makes up human cultural systems. As Dennett pointed out, the term was coined by Richard Dawkins in his book, The Selfish Gene, in 1976. As evidence that mimetic evolution kicked organization up another level, Dennett cited the calculations of Paul MacReady, you'll remember. At the dawn of agriculture, humans and our domesticated animals constituted but 0.1% of the terrestrial vertebrate biomass. Today, we constitute 98% of that biomass with our domesticates. Another stunning transformation, says Dennett, and this one a product of the Cultural Revolution, on a par with the eukaryotic revolution, the parallel he was making. Who designed these cultural things, these innovations that made that transformation possible, Dennett asked? Sometimes we know. For example, just as Dennett coined the phrase Darwin's dangerous idea, Dawkins coined the term meme, sometimes we know who the author is. But sometimes it's just the cumulative product of differential replication in society. As examples, Dan mentioned the decimal system, money, maps. They may even evolve gradually, like making a straight edge, as he talked about, in a way that nicely parallels genetic or organic evolution. But there is also a new emergent feature in this cultural track, We represent our reasons for things, which of course gives us a lot to fight about in society in general, and especially in election years. Dan's was an excellent and provocative presentation, but it seemed to me a bit thin on the intellectual history of cultural evolution. Of course, one can't do everything in a 50-minute presentation. But when coming here to Stanford, where the construction of today's culture evolution theory literally began, I would have thought some sort of modest acknowledgement would have been in order. I'm referring to early path-breaking work here by my colleagues Luca Cavalli-Sforza in 1971, joined by Mark Feldman, in a series of classic papers from the early 70s, starting in 73. Then their big book, Cultural Transmission and Evolution in 1981, and eventually by more of us in anthropology in the 1980s and the 1990s. But that's a small point. And in any case, we have another chance to rectify matters when, in just 2 weeks, Professor Melissa Brown of Stanford will begin to talk about evolution and human societies. All told, then, Dennett opened up a new meme's eye view of the world to us and made for interesting discussion in the panel and again at lunch on Tuesday. Dennett emphasized first that memes are products of the brains and they reside in brains. Because brains are different from one person to the next, the topic came up at Tuesday lunch, aren't there then many versions of a given meme? If the brains are different, won't there be many versions of the meme? Again, the, quote, God meme came up as an example, the concept of God. Dennett confirmed that indeed this was the case, that some memes are harder to fit into some brains than others. No matter how hard he tries, he said, he cannot understand quantum physics. The second, the many versions problem, also raises the question of fidelity or stability of the replication in culture. It might seem that no two people would ever have entirely the same meme in mind. How could there be transmission over time? How could there be evolution without faithful replication? A criticism the French scholar Dan Sperber has made of meme theories. At lunch, Dennett came out with a lovely expression, mundify the epigastrium. Though few of us could understand it, we could all repeat it exactly, which Dennett said shows, mundify the epigastrium, shows that information gets digitized by culture via systems of norms that help us build the common understandings on which culture depends. I thought it was a brilliant example. One last provocative notion that Dan concluded with. He said that music and the arts may have evolved to stimulate, quote, circuits in the mind and thus, quote, tweak the systems of the brain to produce pleasure. He suggested that a music concert, from this perspective, might be a professional stimulator of such brain systems. He suggested also that susceptibility to religion might have functioned as a kind of prehistoric health insurance to guarantee at least placebo effects, if not stronger effects, from non-Western healing ceremonies. A question that Dennett did not answer is, where do new cultures or languages come from? How do new cultural species form, to make explicit the metaphor? No doubt he wishes he were with us tonight, as we now turn to our speakers for the evening for a fresh look at how and why species multiply. Please remember there is no lunch tomorrow. The lunch with the Grants was today. But do remember that we return to the Tuesday lunch schedule next week when Professor Niles Eldridge, Curator of Paleontology at the American Museum of Natural History, will be with us for Tuesday lunch. Sign-ups with Joe just outside. At this time, I welcome to the podium Professor Carol Boggs, member of the Faculty Planning Committee of this class, Director of Stanford's illustrious program in Human Biology. And I'm pleased to announce, I think for the first time publicly, that the Stanford trustees announced this week the establishment of the endowed Bing Director of Human Biology, and it is certainly a lovely honor for Carol to be the first holder of the Bing Directorship in Human Biology. I give you Carol Boggs.
It's an immense pleasure to be able to introduce Peter and Rosemary Grant tonight, who I've known for a number of years now, off and on, having met them both here at Stanford as well as in Colorado at the field station where I work in the summertime. They both got their undergraduate degrees in different universities, Rosemary in Edinburgh, and pardon my pronunciation, and Peter at Cambridge. And then they met at the University of British Columbia, and from then on out, their paths have been very much intertwined with each other, and so have their CVs as well as their interests. Peter did his PhD at the University of British Columbia. Rosemary did hers in Uppsala University in Stockholm. From the University of British Columbia, they moved to McGill University, thence to the University of Michigan, finally settling at Princeton University where they are today. They told me that as of September 1, they're now emeritus. They're not really sure that was the world's best idea, but nonetheless, this gives them more time for doing things like coming to talk to us, which we very much appreciate. They've won a huge number of awards between them over the years, including a number of fellows of a variety of scientific societies, including the Royal Society of Canada, the Royal Society of London, the National Academy of Sciences of the US. They've also won a number of prizes, including the Balzan Prize for Population Biology, the E.O. Wilson Prize from the American Society of Naturalists, and a number of honorary degrees, including from an Ecuadorian university in Quito, which I think is quite an honor as well, given that much of their work has been done in the Galapagos Islands. Now, I tend to think of Peter and Rosemary Grant as people who work on Galapagos finches, which is what they're going to tell us about tonight. But when I looked back at Peter's CV, he started out life working on birds, but he took this detour through mammals. So he's got some diversity in terms of his background that comes to inform as well some of the experiences that they've had in the Galapagos. And tonight what they're going to do is to tell us a little bit about the cumulative work that they've done in the Galapagos, which is really seminal in terms of our understanding of speciation, in terms of our understanding of Darwinian biological evolution, and a talk on how and why species multiply. Now, this is a tag team event, and I believe that Peter is beginning first. Is this correct? Okay, so I give you Peter Grant.
Thank you very much for that introduction, and also for the invitation to both of us to come here and give this talk in a very exciting series. So many evolutionary biologists today follow in the footsteps of Charles Darwin by asking the question, why is it that the world is so full of species, and why are so many species different from each other in so many ways? Well, that question could be asked in a variety of ways, either by looking at things as different as viruses and, let's say, fish, on the one hand, or comparing organisms within a single family, such as these birds on Madagascar, and these ones in Hawaii, honey creepers. The problem of trying to explain in evolutionary terms how a single species has diversified into many of such different types can really be reduced to the essence problem, as I call it, of explaining how two species are derived from one. And this little depiction on the screen shows the problem, the process rather, in outline and cartoon form. What you should do is see this as a change through time of a single lineage that splits into two. These two populations diverge in morphology, and there comes a point, sometime in the future, when they are so different that they are incapable of exchanging genes. They cannot interbreed even if they attempt to do so, and they are, at this point, undoubtedly two species. And the problem for us evolutionary biologists, and it was one for Darwin as well, is to explain that process, multiplied over many times to give rise to all of the diversity in the world that we have. So I'm going to start with Darwin with a quote that gives an idea of the way he thought about the process. Those cases, he wrote, in which a species splits into 2 or 3 or more new species, I should think near perfect separation would greatly aid in their specification, to coin a new word. Well, fortunately, that new word didn't catch on, but the fundamentally important process of isolation of lineages diverging has persisted with us today. But beyond that, Darwin, apart from giving us a theory of evolution by natural selection, which in itself is an enormous contribution. By the choice of words, I don't mean to diminish the importance of it. But in addition to doing that, he sketched out a form of explanation for speciation, but without giving us the detail as how he envisaged the process to unravel. And we have to turn to a predecessor of his, the geologist Leopold von Buch, to get such a description of how the process is thought to have occurred. And so here's a fairly long quote, and it goes as follows. The individuals of a species, he wrote, are spread out over the continents, move to far distant places, that means they're geographically isolated, form varieties on account of the localities of the food and the soil, meaning environmental factors, which owing to their segregation in space, cannot interbreed with other varieties and thus be returned to the original type. Finally, these varieties become constant and turn into separate species. Later, they may reach again the range of other varieties which have changed in like manner, and the two will now no longer cross, and thus they behave as two very different species. Well, the question raised by this is how did this state of no longer crossing arise, and why? Well, let's fast forward more than 100 years into the realm of genetics, and here is a geneticist giving a simple answer to that question. Thus, a long period of non-mixing of two groups, wrote Muller, is inevitably attended by the origination of actual immiscibility, that is to say, genetic isolation by random processes, random genetic drift. That's the short answer of how that process unfolds. The long answer is much more interesting, and it's one that Rosemary and I have been seeking by studying Darwin's finches on the Galapagos Islands for more than 30 years. This is a very convenient group of organisms for studying that question for a number of reasons. The radiation of finches from a common ancestor took place in a relatively short time of 2 to 3 million years. And it resulted in 14 species adapted to different ecological niches, as illustrated by the figures around the side. Now, more than that, these 14 species all exist today. None have become extinct as a result of human activity. And one can't say the same about many other groups of organisms of similar evolutionary interest. And in addition, some of them occur on some islands which are in as close to the pristine state as you could wish for. Which means to say that whatever we can find out about the relationship between organisms and the environment is as close as we can get to the actual circumstances in the past that gave rise to the evolutionary diversification whose products we see today. So here's the outline of our talk. First of all, I'm going to be introducing you to the diversity of species, but very briefly. I'll then, for reasons that become obvious, talk about genes affecting beaks of these birds. I'll outline a theory of speciation and how the group radiated, give you evidence for evolution occurring when the environment changes, which we are able to witness. And then Rosemary will take over, and she will discuss with you what we know about the reproductive barrier between species, the barrier that prevents them from interbreeding. That barrier is not complete. As you see from the last item here, hybridization does occur, and Rosemary will give you the current understanding of why it happens, how it happens, and what the consequences are. It's a rare event, but it could be of great significance. So first, the variety. Perhaps the most famous of all of the species is the woodpecker finch, because it uses a tool for gathering food, not gathering, but prying food out of crevices in bark. That tool is fashioned by itself, but the beak which does it is itself a tool. It's a tool for gathering and dealing with foods. And as I go through a series of pictures of these finches, take a look at the beaks in particular. Color and pattern of the plumage doesn't vary very much, but the beaks certainly do. So here is the Warbler Finch, for example, a little bird of about 8 grams that has a thin needle-like beak, very similar to warblers. It's true that there's a little bit of color in this one, but not in the others. Now compare the beak of this one with the beak of the large tree finch. A much different tool, suitable for crushing, as it is used, for crushing twigs and even small branches to get at hidden arthropod larvae. And then once again, that is different from the beak of the cactus finch, which is a tool used for probing Opuntia prickly pear cactus flowers and extracting nectar and pollen. So there's the diversity, and the question of how it arose in evolution is like a two-sided coin. There are two aspects to that question of how the evolution is to be accounted for. For one side of the coin, one needs to go into the development of the beaks of finches to understand what the factors are that differ between the species in order to be able to infer how evolution has happened. And the other side of the coin is the environmental one. Outside the finch's beaks, the circumstances favoring or fostering the proliferation of species or the divergence of one species into two. So I'm going to be talking first of all about what we know concerning the genetic control over development of beaks that differs between species. And in so doing, I'm going to be talking about the collaborative work that we have done with Cliff Tabin and Arkat Abshanov from Harvard University. These two are the professional molecular geneticists. We're not. We've collaborated with them, but they are the ones who do the lab work. And what they have done to find out how it is that beaks are developed so differently in the species is to take as a starting point a list of the genes that are known in vertebrate craniofacial development, particularly from mammals, including ourselves. There's quite a bit of genetic knowledge of how our faces and crania develop in embryonic form. So we gathered data for from these finches, and our colleagues tested several of these candidate molecules, and they found one, a member of the bone morphogenetic protein family, of considerable interest because it varied very much in its expression across an array of beak types. Now, as you look at the screen, notice the Magnorostris, the large ground finch, is very different in beak size from Scandens, the cactus finch, and there are other species in between these two extremes. The middle panel shows a slice, vertical slice, through the upper mandible only of these two species at day 5 in their embryonic development inside the egg. The egg develops for about 12 days. And the black staining indicates the expression of this particular gene. And in short, in the large species with deep beaks and wide beaks, this gene is expressed more intensely over a broader spatial domain and earlier than in the other species. And there's a graded nature of expression between the extremely large bird at the top and the very needle-like Scandens at the bottom. So that association or correlation between the expression of a gene and the final phenotype that is developed, the beak size that is developed, suggests, but by no means demonstrates, that this gene makes a difference between the big beaked birds and the small beaked ones. So to test this functionally, our colleagues used the clever technique of being able to inject a Magnorostris gene with the appropriate tissues inside the mesenchyme of the developing beak of a bird that doesn't express it like that. Their subject was the chicken, a chick. So at the same age, the gene was injected with a retroviral vector into the same tissues where it produces a big beak in Magna rostris to see if it produces a big beak in a chicken. And it did. The middle panel shows a rather grotesque chicken at the same age that has been treated with the Magna rostris BMP4 gene. The top one, and look at the double-headed arrow to appreciate the difference, is normal. That's the control. That's the one that has not been injected with this gene. It's growing under normal circumstance. And the bottom bizarre creature has been given a treatment of another gene called noggin that knocks out or antagonizes the influence of the bone morphogenetic protein gene. It neutralizes or effectively destroys its action, and in so doing destroys the development of the beak. So there is the evidence that this one gene, BMP4, makes an important difference on how deep and wide the beak of a species is, according to its expression. Well, what about length? The length dimension is also important in some of these finches. And the next question was, what are the genetic factor or factors involved in that? They did a search and found one. Calmodulin is the name of it, and it's important in calcium signaling in the development of the beaks of these finches, particularly in the bottom two, the cactus finch and the large cactus finch, which you can see in the appropriate panels. They did the same kind of misexpression experiment to make sure that there really is a functional effect of this gene, and sure enough, they were able to establish that it does have this influence at about the same time, interestingly, day 5, but independent of BMP4 expression. So two factors make a big difference in beak shape, acting at about the same time, but independent of each other. Well, that's very exciting. I'm sure there are many other genes as well, but here are two demonstrably important ones, and we now have the task of trying to find out how one species is transformed into another in such a way that the expression of one or both of these genes undergoes some transformation. And that implies that there's variation within a species that then becomes transformed into a difference between a species. In what? Either in the genes, we don't think so, but in the genetic control of their expression, the regulators of these genes. And that is where our colleagues are currently searching for variation maybe of evolutionary significance in the transformation of one species into another. So let's now leave the genes of the beak and go into the environment and ask the question of how one species then splits into two that eventually do not interbreed. Here's a little cartoon that captures the essence of the ideas that I outlined at the beginning of Darwin and von Buch. And everything here is hypothetical. We have an ancestral stock arriving through dispersal from the mainland to one island in the archipelago, and then further dispersal around the archipelago, with change taking place in each of the island environments, partly through random changes, probably, but also as a result of an adaptation towards the local environment, because these islands do not have the same food supplies that they offer to the finches. And in this little example, I have a scheme showing that the populations at each step in this cycle of events became progressively smaller. So the crucially important stage of secondary contact between two populations, the size difference had become sufficient that possibly they would not interbreed. That speciation, if that really has happened, speciation would have been complete in geographical isolation. However, our predecessor, David Lack, and we ourselves think that the two are likely to have interacted in a scheme like this, that they would have interacted when they encountered each other, because the differences would not be so profound as to erect a complete barrier to interbreeding. And under those circumstances, we would expect natural selection to favor the biggest of the big and the smallest of the small of these two. And why? Because that would have resulted in a minimization of two things. The first is competition for food. because the more similar the two species are, the more likely it is that they will compete for the same food. And secondly, interbreeding between the two, a minimization of interbreeding between the two, because the offspring produced by the small ones mating with the large ones are likely to be less fit than the offspring produced by small ones breeding with small and large ones breeding with large. Well, Rosemary is going to talk about the reproductive aspects of this process that results in what is called character displacement, a divergence in morphology of the two forms. And I'm going to concentrate on the ecological aspects, starting with not these islands, but islands to the north, Genovesa, Pinta, Wolf, and Darwin. And the species in question I'm going to use for illustrating this is the sharp-beaked ground finch, because if one goes to these islands, one can see the stages illustrated diagrammatically in this depiction. So up in the highlands of the island of Pinta, the sharp-beaked ground finch is to be found occupying a habitat where it exploits snails, arthropods, large and hard seeds, and fruits. Here it is shown it has a sharp beak, as the name implies, but it's also fairly robust. It has a fairly deep beak, even though it is sharp. So there it is, and that in its environment, it feeds on what is available. On the island of Genovesa, in the northeast of the archipelago, the food supply is different. It is morphologically different, and it feeds on what is available with beaks that are appropriate for what are available. And what are available are small seeds and small arthropods and nectar and pollen from these prickly pear cactus flowers. So already you see the association between what is available and the morphology of the birds on different locations. But now when we move to the northwest of the archipelago to the island of Wolf, we encounter another difference that appears to be unique to this population. Here the birds with stronger beaks attempt to break open the eggs of seabirds known as boobies. And they do so by hammering away at the eggshell to see if they can break it open. And if that doesn't work, they use a stratagem as shown here. Kicking backwards, an egg will eventually hit a rock, split open, and then there'll be a free-for-all for the rich supply of protein and moisture on the inside. But that's not all. Interesting as that is, this is even more bizarre. They feed on the blood of the hapless boobies that laid that egg. They do so by jumping onto the backs of the birds that are sitting in the vegetation and reaching to developing wing feathers, pecking at them, drawing blood, and drinking that blood. Now that is a habit that's almost certainly derived from an earlier one of feeding on hippobossid flies, which are little flies that run up inside the feathers of these birds, and much as mosquitoes suck our blood, they suck the blood of the boobies. So what has this bird done but simply shortened the food chain? I'd like you to notice just how strong and murderous that little beak is. It's a very strong instrument for doing something on this particular island which other birds do not do. So once again, there is an association between beak size, beak shape, the food available on these islands, and how the birds exploit that food supply. One can therefore make the argument that adaptation has taken place as a result of natural selection, and as a result of that process, birds are suited to exploiting the foods available in different island locations. Now that is an argument of correlation. It's inferred from what the pattern we see, and it is strengthened much more if we can demonstrate that selection within an island environment operates, changing the nature of the association between the beak size and the food size through time when the environment changes. And that we have been able to do on this little island of Daphne Major in the center of the archipelago. It's an island of about 120 meters high, about 3/4 of a kilometer long, about 1/2 kilometer wide, and it's never been settled by humans, and I think you can see why. So people ask us, what is it like to actually live on the island? And I'm going to make a little a diversion for a moment to give you an idea of what the bedroom looks like in the top left-hand corner, the kitchen on the right, and the analysis we do in the laboratory of seeds to assess the food supply of the finches, bottom right, and the laboratory with our daughter peering down a microscope on the bottom left. Then a couple of pictures of how we handle the birds and how we measure them, how we measure beak depth, for example, that measure that I've mentioned earlier, and how we take a little drop of blood from the brachial vein underneath the wing for later DNA analysis. And how we capture the birds in the first place. We put up mist nets, as is shown in the top left panel. The birds don't see them when they're against dark vegetation, fly in, get caught, and it's an easy matter to take them out and measure them, weigh them, put a unique combination of bands on their legs and let them go. And then we can observe what they do, we can observe how they feed. We know the identity of the bird just by looking at the combination, the unique combination of colored bands on the legs, time, how long it takes to feed on a particular food. And with that information, in 1973, when we started the project, we were able to test the association between beak size and the nature of the foods that the birds ate. And it was, as expected, a very strong association. That is, the big beak birds can feed on big seeds and the small beak birds cannot. They are constrained to feed on small seeds. So the next question for us was a Darwinian one of what is the inheritance of this trait. Without any inheritance, the variation in beak size amongst medium ground finches is of no evolutionary significance. Inheritance is required for evolution to occur. So inheritance is often manifested, it is manifested in the human population, for example, by an association between us as parents and our children. So tall parents tend to produce tall children, and small parents tend to produce small children. And it's as we found out, the same in the medium ground finch world, where parents, here the mid-parent refers to the average of the mother and the father measurements, the parents produce offspring, average of sons and daughters, that resemble each other very strongly in beak depth. The slope of that relationship is a measure of the degree of inheritance. So the trait itself varies and it is inherited, so when the environment changes and natural selection may be expected to occur, there ought to be an evolutionary change in the population. When natural selection did occur 4 years after we began our study, when in the absence of the rains in 1977, the average beak depth of the population increased and kept going until the rains resumed in early 1978 with a regrowth of plants, and the pressure on the finches was off. During this whole period, 85% of the finches had died, and as you see here, that process was differential. Small birds died at a faster rate than large birds, and the reason is indicated by the little diagram in the top left part of this panel that shows tribulus fruits, which are hard and difficult to crack, being the crucial thing that enabled large beak birds to survive better than small beak birds. Well, our result was anticipated by Darwin remarkably closely, and I will read it out. I would now say, he said, in explanation to somebody, exactly what do you mean by natural selection? I would now say that of all birds annually born, some will have a beak a shade longer and some a shade shorter, and that under conditions of habitats of life favoring longer beaks, all the individuals with beaks a little longer would be more apt to survive than those with beaks shorter than average. So back to this diagram, and the fact that natural selection on heritable variation had occurred. there ought to be an evolutionary change from that generation to the next. The next generation is not shown here. And how much change should there be? Well, from the state of affairs in 1976 up to the dotted line in this diagram, because that's an estimate of the average morphology of the offspring when fully grown, produced by the survivors of the drought. And the actual morphological average is indicated by the star there. In other words, our predicted evolutionary change is very closely matched to the amount of evolution that we had observed. Once again, evolution being a change from one generation to the next, and it's measured by the difference between 1976 and the offspring produced in 1978. So evolution by natural selection on heritable variation had occurred really quite dramatically during this period. The next thing we learned about natural selection and evolution occurred several years later. Now, you'll notice this is a rainfall figure that shows an enormous amount of rainfall in 1983. An El Niño event occurred that year of extraordinary severity. But notice that 2 years later, there was a complete drought. And that's where the action was. And I'm going to illustrate this with a couple of pictures, a few pictures. First, showing what part of the island looks like in the dry season, with almost nothing to be seen growing on the ground. Cactus bushes are green, and the trees are leafless. When the rains come, around about January or February of a typical year, the vegetation greens up, and you see in the foreground low plants growing. This is tribulus. This is the plant that produces those large and hard fruits that made a difference between survival and not survival during the drought of 77. And there you see revealed the ways in which the seeds inside those fruits are extracted only by the large-beaked birds. Well, in the El Niño of 83, those low-growing plants were smothered by other plants, and other plants grew upon other plants. Vines grew upon vines, vines grew upon the cactus bushes on the trees at the back, almost smothering and then completely smothering the cactus bushes. And even in the following year, after the rains had stopped, it was possible to see the effects of El Niño with the extraordinary amount of vines draped over the vegetation. In short, what had happened is the island had been converted from a large seed-producing island to a small seed-producing island. And under these altered conditions, the direction of selection had changed. The pendulum of selection had changed from a favoring of large-beaked birds to a favoring of small-beaked birds. Well, now for the long view, and this is where I'm going to end. The long view of selection across years ought to follow a pattern dictated not only by the droughts that occur at intervals, but the preceding conditions, be they very wet and depending upon which particular kinds of foods predominated. This kind of pattern of rainfall then implies that selection oscillates in direction repeatedly and not just once. The diagram on the screen at the moment is more complex than you really need for appreciating that is actually true. G. fortis refers to the medium ground finch, Scandens to the cactus finch, and the panels from top to bottom are body size selection, beak size, and beak shape. And they're coefficients. They are measures of the strength of selection above and below the line of 0, which if there was no selection, you would just see a straight line centered at 0 across these approximately 3 decades. But you don't see that. You see histogram bars above and below the line for both species and all traits. That in turn implies that morphologically, these birds are not constant. They vary, they change from year to year or groups of years to groups of years. And that also is borne out by our data. Same panels, same array of data, but here I'm showing the mean body size, and beak shape across this time period. And to guide the eye, I put in 2 parallel horizontal broken lines to show the 95% confidence limits on the estimate of the size in the first year, 1973, when we had very large samples. Now, if there had been no change across this long period, you should have seen the symbols bouncing up and down within those barriers, but not departing from them. But you don't see that. You see some marked departures, particularly the bottom right-hand panel, which indicates that, remarkably, the birds are not the same as they were when we started our study. Except, and there is one exception here, beak size of Fortis, the medium ground finch, mid-panel on the left. And there it is in a higher, a slightly different way of being displayed. But even this one turns out not to be exceptional, because in the years 2003 and 4, there was a very serious drought, a very severe drought, comparable in magnitude, the one in 1977, when there was a selective favoring of large-beaked birds. So what happened? Did we see a repeated performance? No, we found the opposite. The size, the average beak size of finches dropped lower than it had ever done before and outside those bounds. Well, why? Well, a new factor in the island had occurred, and that was the large ground finch. The large ground finch had immigrated and established a breeding population in the amazing El Nino of 1983. And their numbers at first were too small to have any impact upon the medium ground finch population. But by the time the drought of 2003 and 4 struck, these birds are now to be numbered in their hundreds. Well, they're big birds, and they're very efficient at cracking those large and hard tribular seeds. So they competed with the large members of the medium ground finch, and as a result, the population beak size went down. So I'm going to finish by using a diagram, a picture, to illustrate this main point. Top right then, the Magnerocetus. In the absence of this, under conditions of food stress, where being able to feed on these tribulus fruits made a big difference between survival and not survival, these large beak members of the medium ground finch were at an advantage over the smaller beak members. But in the presence of this one, some 15, 20 years later, whatever it was, then under the same conditions, these were at a disadvantage because they were outcompeted by this one. And now these ones, although they were also dying, were dying at a slower rate, and they had a relative survival advantage. In short, there had been a character displacement, as indicated in that model I gave you earlier, with this species then diverging away from this one, at that interactive phase when two species come together that have previously been separated. Natural selection resulting in evolutionary divergence for ecological reasons. And I now hand over to Rosemary, and the point on the island which is the easiest one to get on.
So, Peter has left us with two questions. First of all, how is genetic variability maintained? And if you can imagine, as he's shown us, that natural selection is oscillating backwards and forwards in different directions, you would expect that the genetic variability would gradually be eroded, but it's not. So the question remains, how is genetic variability maintained? The next question that he's left us with is, what is the barrier to interbreeding between species? If these two species, Fortis and Scandens, are gradually converging on each other, as she showed us, then the question is, what is it that's preventing them from interbreeding with each other? And the answers to these two questions are related to each other, as I'm about to show you. So first is, what is the barrier to interbreeding between closely related species? Well, the very quick answer is song and morphology, but it's more complicated than this. All birds, all of the Darwin's finches on the Galapagos build very similar nests. All Darwin's ground finches are similar in plumage. Males are black and females are brown, and as far as we can tell, they are similar in courtship display. So some time ago, Peter and Loreen Ratcliffe, a graduate student of ours, asked the question, can an individual discriminate on the basis of morphology, body size, and bill shape between their own and another species? And the way they did this was to take 2 museum specimens and put them on either side of a stick, a female Scandens on one side and a female Fortis on the other, and take this into a territory and ask the male, can you tell the difference between a female of your own species and another species? And the answer was a resounding yes. In fact, during the breeding season, when the males are very sort of souped up, they actually went as far as even mating with one of these stuffed museum specimens. So, and always their own species. So we then asked, well, can an individual tell the difference between their own and another species purely on the basis of song without any morphological cues? And we did this by just playing back in a territory. So when we played back Fortis song, Fortis would come in to the loudspeaker, and Scandens would go on feeding. When we played back Scandens song, Scandens would come in, Fortis would go on feeding. And this can be repeated many, many times. And so clearly an individual can discriminate between their own and then other species on the basis of both morphology and song. And it turns out that song is very important between these 2 species. So what do we know about song? Well, first of all, song differs between the species. Scandens sings a ch-ch-ch-ch-ch-ch-ch song, and Fortis we call the Swiss bird because he goes mostly, 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 mostly. And we also know that, well, really they're quite dull birds because each individual has a repertoire of 1 song. And so, but there are several songs along the basis of a mostly mostly theme. So you can have mostly mostly, which is up at the top there, and then you can have mostly mostly sung very quickly, three times or 4 times or even 6 times. But an individual will only sing 1 type. And sometimes little bits of the song are left behind, and it sounds more like a che-ye-ye. So there's individual variation on the species-specific song theme. Only males sing, and we have found no association between beak morphology and song subtype. Exactly the same with Scandens. There's a theme on this ch-ch-ch-ch. The song can become more or less compressed so that you almost get a trill. But once again, we have no association between beak morphology and differences in song subtype. The other thing we know about song is work that was done by Bowman in the 1950s when he was able to bring birds off the Galapagos to California and actually raise them there. And so they had a breeding population of various species of Darwin's finches. You can no longer do this, but he found out some very interesting things. And one is that the song is learnt during a very short sensitive period of time, very early in life, between day 10 after hatching and day 40, which coincides absolutely with the last few days in the nest, and when the birds are out of the nest being fed by their parents, mainly their fathers. So it's not too surprising that a bird learns almost a perfect rendering of its father's song. And a little anecdote with a sample size of 1, which shows how important this is. Early on in the 1970s, a Scandens got a cactus spine through its throat, and it sang a very rusty sort of chir song. And its son learnt this song, absolutely. And even today, we have in the population probably a great-great-great-great-great-grandson of this bird still singing these really rusty sort of Scandens songs, a sort of roughly chir. So once the song is learnt, it is retained for life, and life often lasts as long as 12 years, sometimes as much as 16 years. So this is a typical Lorentzian imprinting-like process, and probably for those of you who don't know what a Lorentzian imprinting-like process is, this was found by Konrad Lorenz many, many years ago when he noticed that the geese if they didn't see their parent as soon as they hatched and they saw him instead, they would think that he was their parent. They would follow him around all over the place, and here's some geese following Konrad Lorenz. And the next year, when the breeding season came round and they got ready to breed, instead of courting another goose, they would court Konrad Lorenz. So this is absolutely what I've just told you is identical to this imprinting process, whereby a species-specific song is learnt very early in life and it influences behavior later in life, such that individuals mate according to species-specific song type. So, of course, imprinting process such as this is vulnerable to disruption if a young bird hears and learns a song of another species during this short sensitive period. And this does happen occasionally, and it happens for the following reason. Sometimes a Scandens, which is a slightly bigger bird, will take over the nest of a fortis, will turf out all the eggs, one is left behind, and so here we have a fortis growing up in a Scandens nest, and it learns its foster father's song. It also happens during the death of a father. The females do not sing, so if a female rears the rest of the offspring, then, or some of the offspring, and the nearest natal neighbor is a bird of another species singing away, it will learn that species song. And it also happens with a heterospecific neighbor. So if there's a fortis and, let's say, a magnorostris nesting very closely together, as sometimes happens, a magnorostris will sing on top of the fortis song and chase the fortis away, and so the birds in both nests will learn a magnorostris song. Now, this happens really quite rarely. It's about 1% or less than 1% of the breeding population. But nevertheless, this rare learning of another species' song has led to hybridization because individuals will mate according to species song type. So the next question is, of these hybrids that are formed, how fit are they? Are they viable or are they fertile? And the hybrids have been formed at this low rate, about 1%, throughout our whole entire study period. The rate has not increased at all. But for the first 10 years of the study period, none of the hybrids survived to breed. They didn't survive the dry season. And so we thought, well, maybe this was because of genetic incompatibility at this time, because fortis and Scandens are not each other's closest relatives. But there was another reason, if you can remember from Peter's talk, that for the first 10 years of our study bird, the seed bank was made up of mostly large hard seeds, tribulus seeds, and also Apuntia seeds. The large fortis ate the tribulus seeds, and the Scandens ate the Apuntia seeds. But hybrids have intermediate beak sizes between these two parental species. We saw them try to crack tribulus, but we never saw them being successful. And we measured the time they took to eat Apuntia seeds, and they took about 2 to 3 times longer to crack Apuntia seeds than they did, than Scandens did to crack Apuntia seeds. So this seems to have been the reason, because after 1983, when small seeds were really abundant in the seed bank, then the hybrids began to survive, and they survived extremely well. So we can ask again, how well did the hybrids survive relative to the parental species? So what we did is we took all those birds born in 1983 and 1987 and 1991. These were years when we had large numbers of species being produced, and so we had a decent sample of hybrids as well as the parental species. And you notice that in the dry years, the droughts that occur here and here, actually the birds don't even come into breeding condition. They just don't breed in those years. So for these 3 cohorts, 1983, 1987, and 1991, this is the survival curve of the parental species. And if there was any disadvantage, genetic incompatibility at all, between the hybrids, we would expect the survival in each of the cohorts to lie below the survival curves of the pure species. And instead, this is what we found, that hybrid survival was as good as, if not better, as the pure species. Not only did they survive very well, but they also produced as many eggs, as many nestlings, and as many fledglings as the pure species. So there seemed to be no genetic incompatibility at all. So how did these birds mate? Well, if we look at this, this is a Fortis scandens hybrid, and this is formed from a Fortis that learned a Scandens song, so sang a Scandens song, mated with a Scandens, and produced this Fortis scandens hybrid, who also sang a Scandens song, mated with a Scandens female to produce the next back cross generation, mated with a Scandens female to produce the third back cross generation. If it was a female, the pattern of mating was exactly the same. Even though the females do not sing, they do actually learn their father's song, as we can tell by their pattern of mating. So if this was a Fortis scandens female, it would not sing, but it would mate with a Scandens male. And so on down the Scandens side, and also the same sort of thing down the Fortis side. So hybrids backcross according to the song type of their father, and we get genes trickling from one species to the other, but the two species are still kept apart by song. So in spite of genes going across, we still get these two groups kept apart by song. So this implies that Fortis and Scandens are converging genetically. And after 1982, when the birds are starting to hybridize and survive. So we can ask, by how much are they converging? So what we did is we took our 16 microsatellite loci and we standardized the difference between the two species to 1 in 1982, which is before hybridization and introgression had started, and just looked at the pattern of introgression across the years, and we found that indeed, this is the Scandens plot now, Scandens is converging genetically on Fortis. And when we did exactly the same with the morphological measurements of beak shape and body size, we found that it almost exactly parallels the increase in similarity in genetic composition. So they are really converging. Another way of looking at it is to look at a plot of beak depth and beak length in 1978 to 1982. Fortis, we can see, is discretely different from Scandens, as you see these two dots. And the lines around I have drawn to show that they are two different, completely different song types as well. But in 1990 to 2003, we can see that Fortis and Scandens have converged genetically, well, morphologically in this case, on each other, but still these two groups are kept apart by song. So does this mean the process of speciation is collapsing? Well, it might be, but not necessarily, because the ecological or the food conditions enabling hybrid survival occur sporadically. Thus, introgression of genes from one species to the other is episodic. And we've set up this state of fission and fusion. So there was fission in the first 10 years of our study, and fusion in the last 20 years. And if we look at either Scandens or Fortis, this is a plot of Scandens, we can see that this fusion has enhanced genetic variation. With the 16 microsatellite loci, the population has become more heterozygous, as we've seen here, and also it has increased in beak shape variation. And so the birds now, the Scandens population now has quite a number of Fortis genes in them, and they are also extremely variable phenotypically. If we translate it into human terms, it's like looking at a 4-foot human to about a 7'8" human. So enormously variable in size and also in beak shape. Now, selection can act on this, and increased variation and allow the tracking of environmental fluctuations through time. So we've answered our first question and it is episodic introgression that explains why the genetic variation is not eroded. So we can then ask, well, when does hybridization not occur? And we have, Peter told you about Magnorostris that had colonized the island in 1983 and built up this huge population. And we have now, or we had, 8 fortis that had imprinted on a Magnorostris song. So instead of singing mostly, they go cheer, cheer. But none of them bred with a Magnorostris. And the question was why? Because actually, Fortis and Magnorostris are sister species. So we might expect that. And also we know that there is the occasional hybridization on Santa Cruz. But on Daphne, Fortis is rather small. It's an 18-gram bird. And Magnorostris is a 36-gram bird. So the difference in size is enormous. Magnorostris is twice the size of Fortis. So it could simply be recognition. When species are very different in size, they do not interbreed with each other. But it could also be harassment, because whenever these poor little Fortis opened their mouths and they went cheer, cheer, a Magnorostris whipped in and just beat the living daylights out of them. Now, we had one Magnorostris male that eventually did get a mate after 9 years of trying, and he got a mate after falling completely silent, and then he mated with his own species. So it seems that if the size difference is small, learning heterotypic song can lead to interbreeding, as we saw with Fortis and Scandens. Scandens is about a 22-gram bird, Fortis an 18-gram bird. But when it is very large, when the size difference is very large, Fortis and, as we saw in Fortis and Magnorostris, then a heterotypic song learning does not lead to interbreeding. So what do we now know about the barrier to interbreeding between species? Well, first of all, we know that there's no genetic incompatibility. And we know that the pre-mating barrier is built from song, which is learnt, morphological differences, which of course are genetic and arise through divergence, and also ecological conditions, food that is inappropriate for hybrid survival. And the barrier leaks when birds learn another species' song, the size difference between the species is small, and the ecological conditions are such that they allow survival of hybrids with intermediate bill size. And in the case I showed you, small soft seeds. We also know that these conditions occur episodically. But the main thing is that the exciting thing about this is that episodic introgression, this refuels the genetic variation on which selection can act. So this is very important. If we're getting this injection of genetic variation on which selection can act, we ask, is this something that we have just seen on Daphne, and we also saw it on Genovesa, or do we see it on other islands in the archipelago? How general is this process? Can we actually see the footprints of hybridization and introgression in the recent past on other islands in the Galapagos archipelago? So we know that species of finches differ morphologically from each other, and they occur together both separately and together on different islands. That is very clear. But we thought that if the transfer of genes really does occur episodically, species A should be genetically more similar at neutral loci, so at microsatellite loci, to closely related species B on the same island than to species B on a different island. That's a bit of a mouthful. So a very simple way of putting it is that Fortis and Scandens, which has had this episodic introgression on Daphne, should be more similar to each other at neutral microsatellite loci than Fortis is to Scandens on any other island. And we did this for all combinations of species of ground finches. And we found that, indeed, when we did this and we looked at all these closely related species on all islands in the archipelago, for the ground, this is a plot for ground finches, we find that indeed most closely related species of ground finches are genetically more similar to a closely related species on the same island than they are to that species on a different island. And when we do the same for the tree finches, we find exactly the same pattern. So clearly, introgressive hybridization has been occurring on all islands in the archipelago between closely related species. So in summary, how and why do species multiply? What have we found? Well, why must be partly due to the environment. A large number of islands with different ecological conditions allow the independent of evolution of populations. How? We now have some understanding of the genetics underlying mean variation between the different species. Peter has shown you that divergence in morphology happens through the tracking of environmental change by natural selection. The pre-mating barrier to interbreeding is built from morphology, song, which is learnt, and ecology. And the barrier leaks occasionally, as I have told you. So introgression occurs episodically, but it occurs only when the ecological conditions allow the survival of hybrids with intermediate beak sizes. So going back to this diagram that Peter showed you at the beginning, the finches lie about here after the lineages or species have become diagnosably different, but long before the point of genetic incompatibility. And I say long before the point because the average time when genetic incompatibility arises in birds is on average 32 million years. And this is work done by Prager and Wilson. Our birds have been radiating for the last 3 million years. And we know that they are still, as I've shown you, they are still genetically compatible. But their lineages are diagnosably different. But if there's introgression at this point, this can be a very rapid route to change. So episodic introgression is a rapid route to change because it augments genetic and phenotypic variation on which selection can then act. It's really quite analogous to an artificial selection process. Darwin started his book on the origin of species describing artificial selection. And this, again, you can think of as analogous to artificial selection process because lineages are crossed after a period of independent selection, resulting in an increase in genetic and phenotypic variation, and then breeders follow this by selection. If it's in crops, it can be selecting for high protein contents or high milk yield in cattle. But how much does it happen actually in the wild? And how general is it in the wild? And if you think about it, it really is very general. If you think about bacteria exchanging DNA and various things like antibiotic resistance has passed over in this way, it's rampant in plants, so much so that Mike Arnold would say we should not think of the tree of life, but the web of life. It occurs in fish, in insects, amphibians, mammals, even in primates, and then I'm thinking of the lemurs in Madagascar. So it is something that I think we should really take into consideration when we are thinking of the evolution. So I would like to thank many graduates, postdoctoral fellows that has helped us, collaborators, assistants, our daughters who have been very important in this, our funding, but anybody who has been to the Galapagos and who has worked on the Galapagos, and many of you have been to the Galapagos here, I think would join me when I say that neither species nor environments are static entities, but they're dynamic and constantly changing. And to conserve species and their environments, we must keep them both capable of further change. And I'd like to, on this theme, I would like to leave Darwin with the last word when he says, it's not the strongest of the species that survive, nor the most intelligent, but the one most responsive to change. Thank you.
Okay, so at this point, I'd like to, after thanking Peter and Rosemary very much yet again for an absolutely stimulating and fascinating talk, to move to the next part of the evening, which is the panel discussion. It will be this evening myself and Professor Rodolfo Dirzo from the biology department, who is also on the planning committee for the course as well. And we'll follow the same format that we've done in previous evenings, where Rodolfo and I will each give some short reflections on the talk that we've heard. We'll give the Grants a chance to respond, should they want, and then we'll open the floor to questions. And I will be moderating those questions again as well. And I think I'd like to take the privilege of starting first.
Ladies first.
And that's one way to say it. And to say that this was really an absolute tour de force of a fantastic and classic example of the operation of evolution, both at the micro and the macro scale in an organism. And I think that it highlights the role very effectively of both understanding the ecology of an organism and a set of organisms, its habitat, its environment, which give you the selective pressures which are needed to understand what's going on in evolutionary terms. But the other thing that was also very helpful and useful is the mechanistic approach to understanding what happens in evolution. Not only understanding the behavior, but also understanding the development, some of the genes that underlie the beak morphology, some of the processes that selection is therefore operating on in order to be able to produce the wonderful variety and the incredible changing patterns that we saw. I think the Grant's work also dramatically illustrates the benefits of long-term and place-based research. You simply cannot do these kinds of studies without having some kind of understanding of what happens over time. Imagine that they had been out there and only looked in 1973, and then the year of funding that they had from McGill University dried up. It was hard to get to the Galapagos. They never went back. We would never see the incredible picture that we saw today. Or even if they were only there for 5 years, the understanding of what happens with changes in environment that occur over a much longer time span to yield that intricate pattern of integration of genes across the two species that drives the speciation, that drives the diversification, would simply not be there at this point. And again, being able to look at something in depth in a particular location, to understand what happens over time, to understand those vines growing up over the cacti, to understand how that results in some of the changes in the seed sizes, is a tremendous advantage to being able to pick apart a system and to understand what happens in both ecological and in evolutionary time in such a system. So this is a classic study of both micro and macro evolution, of what happens both in short-term adaptive patterns, responses, as well as in the longer-term pattern of the development of what happens with different species that builds on and expands on Darwin in the modern context. So Darwin would have probably been very interested in some of the ideas of the reticulate evolution that we saw tonight, with the integration, the hybridization, the passage of genes between these different species that helped maintain the variation for natural selection to act on. But it's certainly nothing that he envisioned when he was writing in this sense. And I think that was very well summarized by the opening lines, where we had species that became different. They weren't immutable, but they became different and differentiated here. So this raises very much the question, I think, of what is a species? And it's something that biologists have debated for quite some time. the complexities of this reticulate evolution, and the fact that you can get hybridization under some ecological conditions, under some behavioral conditions, under some environmental conditions, under some accidents of nature, really poses the question of these units that we kind of lean on in order to understand biodiversity. What do they really mean, and how do we really go about quantifying biodiversity if life is this complicated? And how do we keep track of this? I think Rosemary ended very nicely with what proportion of time life undergoes this kind of evolution. It's something that often you think of as only bacteria would do something like that. But the diversity of organisms that show this, particularly with plants, I think makes the question of what is a species and how does this relate to biodiversity even that much more important. And the last thing that it brings up is, again, something that was brought up in Rosemary's last slides, and that is the issue of climate change. We tend to think of climate change as being something that is a change in the mean. It gets warmer, or it gets higher sea level, or there's a change in the snowfall, or a change in the average precipitation rate in a particular given area. But that's not all there is to climate change. There's also good evidence at this point that this is going to be accompanied by changes in the degree of variation in climate, the frequency of occurrence of extreme events, and that this kind of change will magnify the events of El Niño and La Niña, magnifying the kinds of events that Rosemary and Peter have seen on the Galapagos over time, with consequent potential effects on what happens to the biodiversity there, to the patterns of reticulate evolution, which are a rather unintended and quite unanticipated, potentially, outcome of the effects that we're having on our environment, which will indirectly come to influence even the pristine areas of Daphne Major Island. So I'll leave those thoughts there and ask Rodolfo if he would like to continue.
Thank you, Carol. So I want to thank you again for this wonderful lecture. And I think we all have been benefited and delighted by hearing this great overview of Professors Peter and Rosemary Grant's 30-plus years of work on Darwin's finches in Galapagos, placed in the context of the theory of what is known from the work of other researchers, recent and past, including prominently David Lack and, of course, Charles Darwin. Among the many aspects of this rich lecture that can be commented upon, I have chosen 4 to share with you. One is to say something about what characterizes the work on the Darwin's finches, some of which was presented in this lecture. Second one is the conceptual richness of the book that they describe for us tonight. And I wanted to highlight one aspect, which is biotic interactions as an evolutionary force. Then I would like to say something about Darwin and Wallace. I think we need to pay attention to Wallace as well. And finally, I would like to say something about some possible lessons for conservation, a little bit along the lines of what Carol has just said. So going to my first point, what characterizes the work summarized in this lecture? I think this work helps us to understand how and why species multiply, using Darwin's finches as the model system, through an exquisite blend of the following: natural history, masterful knowledge of the study system, from the history and genesis of the archipelago to the exquisite details of the life of those neo-dinosaurs, the finches. Second, long-term observations in the field, coupled with experimental manipulations to the extent possible given the character of this national park. You saw some of the beautiful experiments with those stuffed animals doing crazy things. Then, of course, is the use of conceptual frameworks to guide the questions, and also the wonderful description that they presented to us in terms of the interdisciplinary and collaborative work, as, for example, the case of the songs. And finally, the application of various tools, technical and conceptual, including those of molecular biology, developmental biology, and song analysis, as well as, and I think this is very important, the retrospective use of current ecological information to interpret and understand the past, which is evolution. Much of our knowledge on evolutionary ecology today is derived from information arising from several sources, different species or lineages studied in different locations and ecological settings, and then trying to paste all of that into a general view. And I think that's a valuable approach. However, the beauty of the work with the Darwin finches that we heard tonight is that the construction of the story of how and why species multiply is based on the same ecological theater, the Galapagos Islands, or the Galapagos Archipelago, and actors that perform the same evolutionary play. My point number 2 is the conceptual richness of the work and an aspect that I wanted to highlight, which is the biotic interactions as an evolutionary force. As you heard, the research on Darwin's finches reveals a still incomplete but fascinating story of evolutionary ecology, as we have seen dating back to about 3 million years, when a group of finch-like birds arrived in the archipelago. This group of colonizers split into 2 initial groups, and this initial diversification was followed by another pulse of diversification, leading to the exploitation of an impressive diversity of trophic niches, from seed eating to ectoparasite feeding, to blood consumption, to lizard tail consumption, to the evolution of the current, leading finally to the evolution of the current 14 species, which results of repeated cycles of speciation, including an allopatric phase of divergence, followed by an interactive phase of in sympatry, with rare but not necessarily zero, and very important as we saw, pulses of hybridization, and in which the allopatric differences were enhanced by selection in sympatry, resulting in character displacements, beautiful examples that we saw today. Over geological timescales, obviously, the ecological theater of Galapagos has been extremely dynamic, giving opportunities for new types of species to evolve. And in ecological timescales, the ecological theory has also changed, and this has given wonderful opportunities to observe and document character displacement and natural selection in action. This is where I would like to reflect for one moment. In particular, I would like to highlight how the evolutionary play is determined by the combination of geophysical and the biotic forces of the environment. And this is beautifully illustrated by one of my heroes of the evolutionary play, G. Fortis. And I hope that this is one of your heroes, as it was highlighted so much tonight. So it's G. Fortis and its changes in morphology in the absence and presence of G. magnirostris, the big guy with the big beak that displaced the other guy. This is a powerful competitor, and it had an influence as mediated by the availability of seeds, those famous tribulus seeds. So as you saw, the average beak size of Fortis became larger in the absence of magnirostris in 1977, and then became smaller in the presence of magnirostris in 2004. If one were to look at these species in 1977 and 2004 without knowing the story, one could have been misled to think that one is in fact dealing with 2 morphologically distinct species. And these dramatic changes in the morphology of Fortis in just 30 years demonstrate the power of biotic forces, competitive interactions, and food plant traits in shaping the morphology of a species, and how biotic interactions are at least partly responsible for the differences we see in coexisting related species. Emphasizing the role of biotic forces might not be newsworthy among animal evolutionary ecologists, but it's especially critical in the case of plant ecologists, who for many decades had followed a tradition of studying plant adaptation and local differentiation in response to the physical factor of the environment, particularly soils and climate. The study of the ecological and evolutionary impact of biotic forces operating within the same habitat of a given plant population has been neglected for decades. And this has been so despite the great intellectual legacy of Charles Darwin, who emphasized the role of competitors and animals in determining whether the plants in a population will live or die, or whether they will leave more or less descendants. Here's an example of his emphasis, quoted from The Origin of Species. Quote, "If we wish in imagination to give the plant the power of increasing in numbers, we should have to give it some advantage over its competitors or over the animals which prey on it." In the case of the Darwin's finches, we see the impact of plant traits, such as seed size and hardness, in determining the results of the competitive interactions among finches, and we also see the evolutionary trajectories that these animals follow. It will be fascinating, I think, to investigate to what extent the constellation of species and populations of finches in the archipelago operates as an important ecological and evolutionary force for plants in the population biology sense that Darwin indicated in the quotation I just read. And it would be great to investigate also what is the potential for reciprocating selection between plants and birds in the archipelago, that is to say, coevolution. Interestingly, in the chapter on reconstructing the radiation, in the context of adaptive landscapes and adaptive peaks, Professors Grant discussed that the resources that determine the adaptive peaks are not necessarily independent of their finch consumers, and that, quote, consumers and the resources co-evolve. And they refer to the case of the particular large form of Magnus rostris, the big guy, in Santa Cruz and Floreana, where the seeds of one of the food plants, the Opuntia cactus, are exceptionally large and hard, and they suggest, quote, that the evolution of large seed size as a result of directional evolutionary pressure exerted by the finches would have fed back on the finches, resulting in a selective favoring of large finches that could crack them. As I said, it would be fascinating to study in detail such biotic interactions and coevolutionary processes between plants and animals and these and other ecological theaters. My third point, Darwin and Wallace. In the context of the biotic forces of the environment, it is in this context of the biotic forces of the environment that I would like to make reference to the apparent contrast of emphasis regarding evolutionary forces between Darwin and Wallace. In The Origin of Species, Darwin places strong but not exclusive emphasis on biotic interactions that arise from population pressures. According to John Harper, the most prominent promoter of the Darwinian approach to plant population biology, Darwin's emphasis contrasts with that of Wallace, who saw natural selection more as a struggle against the physical forces of the environment. In his discussion of the contrast in emphasis between Darwin and Wallace, John Harper says, quote, organisms form part of each other's environment. A change in the nature of any one component changes the environment and so changes the selective forces acting on others, and there is further feedback in turn as they change. The fact that organisms living in different places are different is relatively easy to explain by Wallacean forces. The question of how so many sorts of organisms are able to persist together in the same place is much more difficult to answer, and it demands biotic interpretation and a Darwinian solution. I hope that increasingly plant evolutionary ecologists will emulate studies like the one of the Darwin's finches described in tonight's lecture, paying attention to the complex interplay of abiotic and biotic forces in shaping organic evolution, and I hope that plant ecologists will pay more attention to such Darwinian intellectual legacy. And now to my last point, lessons for conservation. This is another aspect that I wish to emphasize as a message resulting from tonight's lecture, and has to do with the implications of the work on Darwin's finches for biodiversity science and conservation biology, an aspect that Carol also has mentioned. And I'd like to mention just 3 issues, all related to anthropogenic impact on natural systems. First of all is the risk of anthropogenic impacts on the archipelago. Like in many other parts of the world, the Galapagos faces the risk of habitat destruction, the impact of invasive exotic species. We all have nightmares about the potential impact of weeds, rats, goats, and perhaps the less obvious but dreadful possibility of Cactoblastis cactorum, the cactus moth, somehow reaching the archipelago. This cactus moth is a famous moth that was imported from Argentina to Australia to get rid of the Opuntia cactus, and now we know that it's coming back. It's already in Florida, and of course we Mexicans are terrified with the possibility that this cactus moth might make it to Mexico to destroy this diversity of Cactaceae, but I also have nightmares that somehow these little guys might make it to the archipelago. My second point about anthropogenic impacts is climatic change, of course, and the impacts on the species populations and their biological interactions. Given the amazing baseline information available, the Galapagos archipelago can, ironically, serve as an ideal laboratory to assess the ecological and evolutionary consequences of natural and anthropogenically driven climatic change. The factors that could be important to assess, as Carol mentioned, include, amongst those, the frequency of ENSO, atmospheric CO2 enrichment, changes in foliage quality and defenses, with potentially cascading effects on insects and birds. And finally, our preoccupation with biological extinction. So the standard preoccupation in conservation biology is obviously the extinction of species, but we come back to the question, what is a species? And I think we need to pay attention to the extinction of populations, as this is surely massive and represents the loss of equally important evolutionary units. Finally, I'd like to draw your attention to the closing remarks of the book, as they provide an important message to all ecologists and evolutionary biologists. As an ecologist myself, I am always concerned and even frightened when, for example, using my hat of conservation biologist, don't see an obvious connection of that work, my work on conservation biology, with evolution. And I remember the warning that Theodosius Dobzhansky made in his famous quote of 1964. Nothing in biology makes sense except in the light of evolution. In the last paragraph of the last page of the book, Professors Grant say that for the subject of the book, which is how and why species multiply, it could also be said that nothing in evolutionary biology makes sense except in the light of ecology. And then they conclude, this is a good reason for hoping that the light provided by ecology will neither dim or go out. I think this is a powerful and stimulating closing thought, appropriate for a powerful and stimulating book. To put it in eco-evo terms, the cross-fertilization between ecology and evolutionary biology can produce a terrific intellectual hybrid, and indeed back-crossings of high intellectual fitness, as shown in tonight's lecture and in this lovely book. Thanks again for the lecture. What a wonderful way of celebrating Darwin in this and his indelible legacy. Thanks.
Okay, so if either of you would like to briefly respond, or we can go to the floor for questions.
I'd like to hear, Carol, what your view about a species is.
Well, I was, of course, raised on a biological species concept. And so that would mean that things that are not interbreeding, whether it be for pre-mating, isolation, or otherwise, are in some senses, in my mind, good species. But as a conservation biologist, I also worry about diversity within a species. And so maintaining the processes that can lead to hybridization, that can lead to reticulate evolution, that can lead to the maintenance of heterogeneity within a species are also important, would be my answer.
So I think it's appropriate for either Rosemary or myself to say something about what we think a species is, because everybody wants to know that. We work with species, we ask how they evolve, and how a new one is formed. We'd better be prepared to say what we have in mind when we use the term species. Well, of course, it's not a simple matter. It used to be, when it was thought that all one needed to do was to find out whether two forms, two populations, if they attempted to interbreed, could not do so because any offspring that were initially formed in embryo would die. Or the offspring grew up, so they were viable, but they were infertile. They could not breed. Because that meant that each of the populations was incapable of exchanging genes with the other one. The barrier to breeding, to exchange of genes rather, was complete. But now we know from the last 70 years of research in particular that there are all sorts of gradations between that state and the state where there may be a 2 or 3% loss in fitness in members of population A breeding with members of population B. Darwin more or less made the same point, that species and varieties, in his language, were not categorically separable in any objective way. I think modern biological research has backed him up and showed that to be the case. And so it almost comes down to the question of by how much, if one wishes to adopt a biological species concept, that is a concept of a species as a biological unit of interbreeding individuals, of being animals or plants, incapable or rarely capable of breeding with something else, one has to come up with some kind of criterion for saying this is where the line is drawn between two entities that we're going to consider species and two that we're going to consider well-marked subspecies. I wish I could remember exactly how it was stated, but the British Ornithologists Union was charged with the same task for advising the members of their society on how species are recognized. Our book quotes them, quotes a committee's results. Now, I wish I could remember the wording, but it goes something like this. If two populations occur together in the same environment and they do not interbreed, or if they do, they produce offspring that are relatively unfit, or if the two populations produce fairly fit offspring, but the two populations do not merge into each other as a result of that interbreeding, then it is profitable to consider them as species. They are essentially, if not functionally completely independent, they are functioning as two independent units. Then there comes the question of two populations that are in different environments. They look very different. And a systematist looking at the morphology might be inclined to say these are two species, or might be inclined to say that they're two marked varieties of the same species. By what criterion are we going to establish that, a basis of classification? And their answer, the committee's answer, was the only way of doing that is to compare the differences of coexisting populations that do not or very rarely exchange genes with the characteristics of those separated populations that are incapable of exchanging genes because they're living in separate locations. Now, there's a very complicated way around the problem, and there is a major alternative which is used by people who use molecular biology to reconstruct the history of species through association of basically their genetic similarity. And if it can be shown that two species are different in a way described as reciprocally monophyletic, which means to say that they have independent origins and they are discretely different, at least in some trait in their molecular genomes, which indicates that they're not exchanging genes, then they should be considered species. There are many difficulties with both of these concepts, and I think our bottom line is it depends what we as biologists wish to use the concept for. That will determine exactly which of those two is the preferable one and how we're going to apply them. That's a very complicated answer to a very short question. And that, I think, uses up all of our time. So thank you very much, everybody, for coming.
I think we do have time for one or two questions, though, from the audience. If there are people who wish to come forward to the microphones, and assuming I can see you out there. Okay? Yes?
I think the concept of introgression from very rare hybridization events is fascinating as an accelerant to the process of speciation, but implicit in that idea is that the hybridization is rare and that there's the chance to backcross. But presumably you could postulate that if the level of hybridization was high enough and the hybrids were fit enough, that it would become harder and harder to have that backcrossing process occur. So at what level does introgression cease to be an accelerant to speciation and becomes a retardant?
That's actually a very good question. The Y diagram we showed at the beginning, where we show that in birds anyway, that it can take on average 32 million years to reach complete genetic incompatibility. I would think your answer to the question would lie at what stage do these species we're talking about lie on that Y diagram? Are they really close to the point where they are splitting onto two independent lineages, going off on each independent evolutionary trajectory, or are they nearer the point at which genetic incompatibility is being approached? So I would see that as the sort of answer. Do you want to add anything to that?
I don't really know. It's a difficult question.
Yeah, over here.
I was curious if you could expand on something you said towards the beginning of your talk. When you talk about genetic changes causing evolution, it's easy to see a replicating entity and how that replicating entity could change and cause an evolutionary change. But when you were talking about the beaks, you mentioned that you didn't really think it was the gene that was changing so much as it was how the gene was expressed. And that seems like a much less sort of binary process. And I was curious, how does gene expression fit into the whole concept we have of evolution being driven by replicating entities that mostly copy themselves but sometimes change a little?
Right. We've looked at our colleagues have looked at the structure of the BMP4 gene and the calmodulin gene to a lesser extent. BMP4, let me concentrate on that one, to see if Magnaroceros, for example, has a slightly different structure in its gene to, let's say, Scandens, the cactus finch. They've found no difference so far. It looks as if the genes themselves are the same in all of these species. And that's not too surprising, because something else I haven't told you but will now is that these genes are expressed in different tissues of the body at different stages of development, rather widely. And it's therefore not surprising that this gene with multiple functions is the same across all of the species. So then you have to ask, well, we know that they're expressed differently in time and to some extent in space amongst the species. What governs that difference? And the answer has to be in the way in which the genes are switched on and switched off, what we call the regulatory apparatus. And that gives rise to, presumably, we don't know this for a fact, we don't know exactly what part of the regulatory apparatus varies, but presumably it does vary among the species because the species are different, but it also presumably means there's variation amongst individuals within a species. Selection acts on the adults. We know that, and we do not think there's any force of selection on the beaks inside the eggs. So it's entirely at the adult level. Therefore, you have to think of it, I think, like this. There's variation in the way in which the beaks form amongst individuals in a population. They grow up to assume different adult sizes. The environment says, you, and you can stay, you, and you drop out. In that process, what in fact is happening is the environment is selecting developmental programs that gave rise to these and penalizing the developmental programs that gave rise to those. And by developmental programs, I'm simply referring to the way in which factors unknown control the expression of the BMP4. I hope, is that clear enough?
Yeah. So, ultimately, so you're saying that there are a fixed and finite a number of essentially programs for how the genes are expressed, rather than an infinite gradation. I mean, are they sort of binary in that sense, or do you see the programs for expression of the genes as being something that is where there's a complete gradation of methods in which that occurs, or do we know?
Yeah, I think the subject is probably so complex that there has to be an interaction between the expression of these regulator genes and feedback from the other metabolic processes that are taking place in the cells at the same time. And those other processes will involve impacts of the environment. That means to say the environment external to the embryo in the nurturing fluid, for example, in the egg. I'm guessing that this is going to be an exceedingly complex network of interactions, giving rise to a graded expression of the genes as opposed to this binary one, as you refer to, an either on or off.
Well, or another, if I can just add a little
Please
bit,
do.
another way you can think of it is that they are being expressed, so as Peter said, sort of switched on or off, but these are pathways, biochemical pathways, which sort of feed back. And so you also, the other thing that could be changed is the receptors. So the receptors could be also altered in different individuals. So you can get variation at all stages, both in the expression, the timing of expression, and the receptors that feed back on that.
So what we've done here is we've moved from ecology, behavior, into genetics, and on into the really new and exciting field of evolutionary development, or Evo-Devo as it's known colloquially, which shows the astounding range of the Grant's work as it goes from one part of evolution into another in order to be able to really understand the whole picture. I know there's still a couple of people with questions, but I think at this point we're running out of time, and we need to thank the Grants very much for a wonderful presentation tonight.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.